place which these microtubules attach to the DNA is the kinetochore. And the kinetochore is found in the region of the centromere. The centromere is just a physical joining of your two sister chromatids, your two replicated pieces of DNA. And then there's a special region within that centromere called the kinetochore, and that kinetochore is what actually attaches the microtubules to the DNA. All right, the next thing that's going to happen is metaphase. This is a perfect example of what a metaphase cell is going to look like. At the time of metaphase, we have this spindle apparatus completely arranged, and we have all of the DNA lined up in the middle of the cell. And again, we also have these microtubules that are attached to the kinetochores. And one thing I didn't mention earlier, remember these are two pieces of DNA. They're just kind of stuck together. So these kinetochores, this microtubule will attach to the kinetochore of one sister chromatid, and then the other microtubule will attach to the kinetochore of the other sister chromatid. So by the time we get to anaphase, remember those microtubules are going to shrink back into the centrosome, and that's what's going to pull those sister chromatids apart. And the sister chromatids are being pulled apart because they're attached to these microtubules using these kinetochores. And so in anaphase, again, these microtubules are going to depolymerize. They're going to shrink back into these centrosomes, and then that's what's going to pull those sister chromatids apart. So anaphase is the actual process of pulling the DNA apart and moving the copies of DNA to both sides of the cell. And then finally, the last phase of mitosis is going to be telophase. Telophase is when the cell's starting to get back to normal. So you're regenerating your nuclear envelope. You're also going to begin cytokinesis. So telophase and cytokinesis, they're kind of, again, they happen back to back and almost kind of simultaneously. But the main thing that's happening in telophase is the regeneration of the nucleus or of the nuclear envelope. And then that's going to be followed quickly by cytokinesis, which is the division of the cytoplasm. And so if you look at some images of what all of these look like, here is when we have a cell in G2 phase. And something I didn't mention, every part of the cell cycle, except for M phase, is known as interphase. And so interphase is composed of G1, S phase, and G2. So in other words, a cell is going to be in interphase 90% of the time when it's in the cell cycle. And so anything that is not mitosis is considered interphase. And so here you can see an example of what a cell looks like at interphase. What they've done here is they've stained the DNA with a blue dye, and they've stained these microtubules with a green dye. So wherever you see green, that's microtubules, and wherever you see blue, that's DNA. So before the cell enters M phase, so the DNA's already been replicated at this point because it's already gone through S phase. So here we have a cell in G2. Now in G2, we remember our DNA is existing as this chromatin. And if you take a close look at this, it's just strings and strands and strands of DNA everywhere. And so you still have your nuclear envelope. Here you have your centrosomes right here. Everything's kind of normal. And then once it enters into M phase and goes into mitosis, this is when this DNA is going to start to condense. And so that chromatin is going to be converted into these chromosomes. So in prophase, you can start to see the DNA condense. Also, what's going to happen is you're going to start setting up your spindle apparatus. And so you're going to have your centrosome here and your centrosome here, and these are all the microtubules that we've been talking about. So notice they're not all the way set up on opposite sides of the cell, but this is the point where everything starts getting set up in anticipation of dividing the nucleus. And then after prophase, we have prometaphase. Again, now here's where you can start to see these distinct condensed chromosomes. Also, the nuclear envelope is going to fragment and it's going to disappear, and the purpose of that is so that we can shuffle all of these chromosomes around without being boxed in by a nucleus. And so in prometaphase, the DNA is going to start to condense, and so now what just looks like a mess of DNA over here now actually looks like chromosomes. And so you can see the centrosomes with all the microtubules. So you can see that the spindle apparatus is starting to get more complete. You're having the centrosomes are now going to be at opposite ends of the cell, which you can see here and here. And then again, this is where you're going to be able to see those chromosomes and where the nuclear envelope is going to disappear. Now in metaphase, this is where we have all of the chromosomes line up in the middle of the cell. And so we call this kind of imaginary plane, think of it as like an equator, an imaginary plane, the metaphase plate. So during metaphase, the chromosomes are going to line along the metaphase plate. I said it's just an imaginary line, but you can see even in this cell, you can see all of them aligned up in the middle of the cell. And then you have your spindle apparatus is complete at this point. You have your centrosome here, and then all of these microtubules are now attaching to these sister chromatids. And again, they're attaching at the kinetochore of each chromosome pair. And then during anaphase, this is where these microtubules start to retract, and because the microtubules are attached one to each sister chromatid, that's the force that's physically pulling those two copies of DNA away from one another. And so if you look at it in an actual cell, you can see here's one set of DNA, here's the other set. And so they're literally being pulled to opposite sides of the cell. And again, the reason for that is that these microtubules, they start shrinking, they start depolymerizing. And so they become shorter and they pull their half of the DNA with them. And then after anaphase is complete, you're going to have your DNA in two different sides of the cell. And so this is where telophase and cytokinesis comes in. And again, these two occur almost simultaneously. But cytokinesis has its own spot in the cell cycle, whereas telophase is still part of mitosis. So in telophase, what's going to happen, you're going to get the nuclear envelope start to reform. So now that we've made copies of our DNA, we're going to make the nuclear envelope, and our DNA is going to start decondensing again. So at this point, our chromosomes are going back into the chromatin state, and you are regenerating your nucleus in the process. And then cytokinesis, which is the actual division of the rest of the cell, you're going to see two different types of cytokinesis depending on if it's an animal cell or a plant cell. Here we're looking at animal cells. So when an animal cell undergoes cytokinesis, it's going to create what's called a cleavage furrow. Cleavage in biology means to break something. And so we're using this cleavage furrow that's actually going to help separate these two cells once we've replicated our DNA. And so we'll talk a little bit more about that. So here, if we're looking at the mitotic spindle, let's just draw a big picture of it so we can get an idea of what's going on here. Because the mitotic spindle is the only reason why this whole mitosis process works in the first place. All right, so we've got our cell. This is our one centrosome. This is the other centrosome. And remember those centrosomes, the microtubule organizing center. So these are what make and break down the microtubules. And so from these centrosomes, you have these little centrioles. You have a little pair of centrioles. And these centrioles are only going to be found in animal cells. So, centriole. And the purpose of the centrosome is it's still a part of this, the centriole is still a part of the centrosome, but it's the microtubules that are going to come out of these centrioles. And so once you have your DNA, so we'll just make it again in M phase. So these microtubules are going to extend from these centrioles, and it's going to attach to a kinetochore of one piece of, or one pair of the sister chromatids. And at the same time, out of this centriole down here, these microtubules are going to attach to a different kinetochore on the other copy of DNA. So you have one set of microtubules attaching to one copy of the DNA, and the other microtubule from the other side of the cell is attaching to the other piece of DNA. And so that's going to be the same for all of these. And then again, whenever the cell is undergoing anaphase, because this is attached to this piece of DNA and this is attached to this piece of DNA, when those microtubules contract or get smaller, that's what's physically pulling the two strands of DNA apart from one another. So it's really important that you understand how the mitotic spindle is set up and also how it works. All right, I see a short answer question in your future. All right, questions? Okay. All right, so here we can take another look at the spindle apparatus. So this is an actual close-up image. You can see here you have the centrosome, and the centrioles are in there too. They're smaller, so you're not going to really be able to distinguish them in this picture. But nonetheless, we have the centrosome on both sides. We have the microtubules coming out, and then we have all of our chromosomes aligned at the metaphase plate in metaphase. And so you can see that here, and here's a nice close-up picture of something that represents a kinetochore. And so we know that this is the region of the centromere, but there's a specific region, which is the kinetochore, which is where these microtubules attach to. And so these microtubules, again, once the cell starts to divide, that's going to pull those sister chromatids apart because you have one set of microtubules attached to one copy of DNA and the other set of microtubules attached to the other copy. And so you can see in this image, it's not a very clear image, but here would be the kinetochore. So this is the region that represents these little black dots right here. And so these are the microtubules right here coming out of the kinetochore. And same thing on this end. This is the other side of the DNA. So you're going to have these microtubules attached to both sides of your copied DNA. And so one's going to go to one side and the other is going to go to the other side. Okay, so we mentioned that there were going to be some differences between plant and animal cells when it comes to cytokinesis. In animal cells, we already mentioned this cleavage furrow. So if you want to get a better idea of what a cleavage furrow is, just imagine that you have a balloon and it's full of air, and you take a string and you tie it around the middle of the balloon and you start tightening that string. Well, that balloon is going to start to pinch in until eventually it's going to become two of its own cells. So that's essentially what's happening here in this cleavage furrow. So it starts in the middle and then furrows its way in until you end up with two dependent cells. And so the way that this cleavage furrow basically contracts and gets smaller, you got to imagine it as like a ring around the cells that you're trying to divide. And so that ring is made up of microfilaments. What are microfilaments made up of? Actin, right? And so you have all these actin subunits that are, again, depolymerizing. They're getting smaller and smaller, and that's what's the physical force that's actually starting to pinch the cytoplasm apart from these two cells. And then in plant cells, it's kind of the same idea, except instead of a cleavage furrow, we're going to have what's called a cell plate. And so this cell plate, what's going to happen is you're going to get these little pieces that are eventually going to become the new cell wall. And so you're basically creating a cell wall that's going to end up separating the two cells that just divided. And so you're going to see this cell plate start to form, and eventually it's going to expand until it creates the new cell wall. So we start with one cell, and we end up with two cells separated by a cell wall. And so if you look at the image right here, and it's kind of hard to see, but you can see this picture, this image looks kind of like this. You can see different chunks of the cell wall starting to form, and eventually that will elongate all the way through the length of the cell until you end up with two new plant daughter cells. Questions? Okay, so here, now we talked about mitosis in animal cells. Here is mitosis in plant cells. It's going to be the exact same process. The only thing that's going to differ is cytokinesis. So prophase is going to be the same, prometaphase is going to be the same, all the way down the line. So it gives us a chance to review mitosis again. So in a plant cell, there are 5 stages in mitosis. The first one is prophase. Again, in prophase, you're going to see your chromatin start to condense into chromosomes. And then in prometaphase, this is where you're actually going to start to see these individual chromosomes. Not only that, but you're starting this nuclear envelope is starting to dissolve at this point. So when you see the chromosomes, but they're not arranged in any particular way, and there's no nucleus, that tells you that you're in prometaphase. But if you see a cell that still has its nucleolus present, then that's going to tell you that it's going to be in prophase. And so the DNA is condensing in prophase. We're starting to see chromosomes in prometaphase, and we're starting to get this spindle apparatus set up. And then in metaphase, the spindle apparatus is complete. So you can see the microtubules here. Again, you have the chromosomes lined up in the middle of the cell. And then during anaphase, again, that spindle apparatus is going to make those microtubules contract, and that's what's going to pull them to opposite sides of the cell. And then in telophase, this is where you're going to get the reformation of the nucleus. And then in cytokinesis, you're going to start to get the formation of this cell plate, which will eventually come across the entire width of the cell and become the new cell wall. All right, questions about mitosis? All right. So let's test yourselves and see if you can recognize different cells in the various different stages. All right. What about this guy? We'll start with an easy guy. Let's do this guy. Anaphase. What makes you think it's anaphase? Yeah, because you can see them being pulled apart in opposite directions. So this one would be another example of anaphase right here. So let's see if we can find some more. We got that one, we got that one, we got that one, we got that one. So there's several of them. And so when you actually can see like the tails of the DNA being pulled apart, you know, basically being dragged behind them, that's how you know they're being pulled. So when they're in the act of being pulled, like they are right here and right here and right here, that tells you they're in anaphase. All right, let's do another one. What about this guy? What do you think this guy's in? Which? Yeah, telophase. So you can see the DNA, it's not being dragged anymore, right? It doesn't have its little chromatin tail sticking out. Instead, you have the DNA on the opposite sides of the cell. And it's kind of hard to tell from far away, but from up here I can see the cell plate starting to form right here. And so in telophase, you're just going to have all the DNA on opposite sides, but they're not going to be actively pulling. So what do you think about this guy? What stage do you think he's in?
Anaphase.
He's in anaphase because you can see his little chromatin tails. And I think this guy is the only guy that's in telophase right now. This guy would also be in anaphase. So it looks like out of all of these, this is the only one that's going to be in telophase. All right, what about this one right here? Another easy one, right? Metaphase, because they're all lined up in the middle of the cell. Same thing with this guy right here. You know that the DNA is condensed, and so you can see these individual chromosomes, and they're all going to be lined up in the middle of the cell. Here's another metaphase cell right here. And so that only leaves us with prophase and prometaphase. So which one do you think this is? Prophase, right? Because the DNA is starting to condense, but you're not going to see those individual chromosomes until prometaphase. So this would be an example of prophase. The DNA is starting to condense, but it's not quite there yet. And so after prophase is complete, then you're going to see these individual chromosomes, but they're not